So let me introduce the two speakers for the eustachian tube and transtympanic pathways for drug delivery. Now, is there such a thing as a transtympanic drug delivery? I think so. And the eustachian tube, it clearly leads into the middle ear and the inner ear through the membranes. Now, we know how to reach the eustachian tube, and we all, rhinologists or otologists, we're doing eustachian tube dilation for the last probably almost a decade in treating eustachian tube dysfunction. So it will be very easy to deliver drugs, therapeutics, and other events through the eustachian tube into the middle ear or through the tympanic membrane. So I'm going to show you two short videos. Here's a patient with a severe recurrent chronic sinusitis. You can see the pus draining from the sinuses. And here is the eustachian tube on the right-hand side, and clearly leads into many infections, not only in the nasopharynx, in the throat, and enters the ear. Rhinosinusitis or nasopharyngitis that can be chronic with a glue-like structure or secretions in the back of the nose and inside the eustachian tube. And you can see the amount of thickness that this glue-like mucus that is coming out from the eustachian tube. And I'm trying to clear that thick mucus. So Sujana is going to enlighten us. Go ahead, Dr. Chandrasekhar. Northwell Hofstra used to be at Mount Sinai and a partner at ENT Allergy, which has how many ENT doctors in the group? 100?
Oh, I think we just acquired another 30. So I think we're up to 250 otolaryngologists in New York, New Jersey. We're down to Cherry Hill, New Jersey, which is like a suburb of Philly. Yeah. So we got it all. Okay. So I want to thank you, Dr. Crespi, for putting together a really interesting group of people talking about very interesting things. I think one of the advantages to having a brain like Joe's is that you think outside the silo. And when we think outside the silo, I think we can advance care. So Joe asked me to talk about something that I know pretty well, which is the eustachian tube drug delivery and the role of surfactants. And he told you who I am. So these are my disclosures, and the only one of pertinence is that the surfactant that I'm going to be talking about, I've been working with my team to bring to market with some adjustments here and there, and hopefully I'll be able to share that with you in the future. I also did serve on the American Academy of Otolaryngology balloon dilatation of the eustachian tube clinical consensus statement. So we all understand that eustachian tube dysfunction affects most people. In regular routine airline passengers, not the people with the thick mucus that Joe just showed, but the normal person walking down the street, about 1/5 of adults and 2/5 of children have significant negative middle ear pressure measurable after any airplane flight. And about half of those people have otoscopic evidence of damage to the tympanic membrane. So you can see barotrauma with flying, with scuba diving. You can see, and in Manhattan, you can see it with people who have to take 2 elevators to get to or from their office if they're in very high buildings because they can't manage. You can see acute serous and chronic otitis media. And then as Dr. Crespi said, we can't just keep putting tubes in people, right? So there has to be some other way to deal with this. So many, many years ago, I'm going to tell you that story in a second. So what we see is we see acute and serous otitis media. We see airplane ear. We can see recurrent otitis media with effusion or recurrent acute otitis media. And then we see sort of the bane of the otologist's existence is these eardrums that keep collapsing down. We do a beautiful repair. We put some cartilage, we put some prostheses, and then a year or 2 or 5 or 10 years later, they come because everything's collapsed down again and they're having problems again. So this is a slow motion video of the Eustachian tube from Dennis Poe, and this is showing you that it's not just an open close. There's a really interesting tensor and levator, veli palatini fire, and there's a sinusoidal movement where the entire Eustachian tube opens, and you can look all the way up, and you're probably getting a look up to about the 12 millimeter mark on the ruler below. So there's a lot of length and volume that has to open in the normal Eustachian tube. So current medications don't work at the Eustachian tube. So everyone gives people intranasal steroid sprays, and they just don't reach and they don't do anything if they happen to reach there. And then people are using topical or systemic decongestants or antihistamines, and they definitely don't work. So there is a unmet need to find something that gets to the eustachian tube and works to stick the mucosa so that the tube can open. So Tarabici, you saw this picture with Dr. Crespi's introduction. You see where we put the balloon for balloon dilatation of the eustachian tube. It's in the cartilaginous ear canal, which is, cartilaginous eustachian tube, which is here. But Tarabici and others have shown that, in fact, you can have eustachian tube dysfunction throughout from the protympanum all the way down the bony eustachian tube to the cartilaginous eustachian tube. So even if we do a good job with balloons, we're only solving part of the problem in our patients. This is just a beautiful cross-section drawing from Bob Rubin that Joe Fagali shared with me. And what our goal is to get some medicine that you put in here, have it travel down the nose, have it go to the eustachian tube orifice, have it travel up the eustachian tube orifice and reach the protympanum so that it can take effect. So that's really the challenge that we're looking at. So many years ago, a friend of mine who was an audiology director at New Jersey Medical School at the time said, "Oh, I know this guy. I want you to meet him." And I said, "Okay." So I met this very smart fellow called Alan Mautone, and he unfortunately has passed away, but he was a very dear friend for a long time. He was an outstanding pulmonary physiology PhD. And he said, "Oh, I have this thing that opens up stuck tubes." And I said, "Oh, I have this stuck tube that needs to be opened." So sort of like, peanut butter and chocolate. And so we made a Reese's peanut butter cup of each other. So the idea is the eustachian tube's not working well. Because it's not working well, there's negative pressure built in the middle ear space. That begets its own serous otitis media to form. If it stays in there long enough, it's going to become bacterial or at least viral and have some inflammatory response. But because there's fluid in the middle ear, like this green stuff that we drew, that also, so the tube is blocked, it causes fluid to build up. The presence of the fluid blocks the tube further. What we want to do is have it drain normally as opposed to putting a tube or a hole in the ear drum to have it drain out into the ear canal. So how do we reestablish normal physiologic opening and closing of the eustachian tube along its entire length? So what about surfactant? So surfactant is so named because it's a surface active agent. It's actually just a conglomeration of those words. But surfactant is normally found in the eustachian tube, and the eustachian tube surfactant is different than the lung surfactant that opens your alveoli. In cases of otitis media, surfactant at the eustachian tube is reduced. So we know that there's some correlation. Surfactant proteins A and D exist in normals, and surfactant protein A is upregulated in ears that have otitis media. So initially, there was a group, Nemechek, the last reference at the bottom, looking at nebulized pulmonary surfactant that was either given intratympanically or intranasally It was a bovine or porcine product. It was not a synthetic product. So it had some proteins in it. It had some animal products in it. And when given intranasally, it was a huge quantity that had to be given. And it resulted in some reduction of the passive opening pressure of the eustachian tube and some reduction in middle ear effusion, but it had to be given in very high, very uncomfortable doses. So there are some surfactants that came onto the market, and these are at least irritating to the nasal mucosa, if not actually harmful. They may actually even be toxic to the nasal mucosa, and we heard really eloquently about the olfactory system and the nerve endings in the nasal mucosa earlier today. And so, unfortunately, a couple of these have led to permanent hyposmia or anosmia and have been banned by the FDA. So this is not enough. We don't have enough. So Alan was able to synthesize a completely synthetic surfactant without animal products in it that we could deliver dry. So it didn't feel like you were flooding the nose with a liquid because you were just spraying what it actually feels like a very cool, dry puff of material. When you spray, it doesn't have an odor, it doesn't have a taste to it. Because it is synthetic, there's no proteins and you can't develop resistance to it. And what we were able to show early is that it didn't actually need your nose to do anything, just to be moist. Because if there's a table, let's say a 3-foot table, and the surface is moist, if you sprayed our surfactant at one end, that surfactant would change shape from a clump to a shard and kind of move along the surface of that table that clearly does not have any cilia on it, and get to the other side. So all you need is sort of a moist surface, and then the surfactant can make its way to the back. Well, that's a very nice idea. Did it work? So this initially, our experiments were with dipalmitoyl phosphatidylcholine and cholesterol palmitate, which we thought we needed that as a spreading agent. It turns out that in 2.0 or 3.0, we don't need the cholesterol palmitate, which is very helpful in terms of manufacturing so that you avoid any intermolecular issues with the manufacturing. And this lines up with hydrophilic and hydrophobic ends. And when it does that, they repel each other so that the mucosa then unsticks. So it's a purely mechanical reaction to what we're administering. So our surfactant lowers the surface tension of any liquid lined tube. So I thought, well, maybe it helps at the eustachian tube. So what happens now is that we give steroid sprays because we want to give something because the patient is very uncomfortable. That steroid spray really works in the middle and where the sinus openings are, front and middle of the nose, but doesn't get to the eustachian tube, or by the time it gets there, it has no action. Our surfactant changes shape into those shards and quickly travels along all the way to the back. And the new actuator that our group has developed is really based on some really intense anatomical and radiographic studies and really targets the eustachian tube very well with a very comfortable intranasal anterior nose application. So our first question was, can we affect eustachian tube dysfunction? So can we reduce passive opening pressure of the eustachian tube in normal animals? And this was done in gerbils and mice, and the turbinates are in fact different in gerbils, mice, and chinchilla than they are in mammals, but it's a pretty reasonable group to study it in. And so we built this sort of comprehensive way to test where we did not puncture the tympanic membrane in order to measure passive opening pressure. And what we saw was that we dramatically reduced passive opening pressure of the eustachian tube with just the spray of surfactant. We did it with just the meter dose inhaler, thinking maybe I'm doing a Valsalva to the animal, and that was not happening. So it was the surfactant that was dramatically reducing the passive opening pressure, and we showed this in mice and in gerbils. So then we said, okay, what happens if we give the animal otitis media with effusion? So this is a Klebsiella pneumonia endotoxin. We tested a whole bunch of gerbils. We had no treatment. We had the propellant alone. We looked at the surfactant alone, and then we looked at the surfactant with steroid and the surfactant with phenylephrine. And we looked via tympanometry and via otomicroscopy. And in order to start the experiment, this is sort of a cartoon to show you these grades, but the animal had to be grade 4, maybe 3 to 4 to start, and they had to be grade 0 to 1 for us to consider it to be back to normal. So what we saw was that in the non-treatment groups, the propellant alone group and the surfactant with phenylephrine groups, it took about 16 days or more for effusion to clear in these animals. And in fact, if you gave phenylephrine twice daily, it really dried out the mucosa, so it actually took them a little bit longer. When we gave surfactant alone once or twice daily, which is the blue and the yellow bars, that reduced that 16 days to 10 days and 8 days. When we gave surfactant with steroid once daily, it took 8 days to resolve the effusion. And when we gave surfactant with steroid twice daily, it took about 6 days to resolve the effusion. And the effusions did not return upon withdrawal of the treatment. So what happened was we established normal physiologic opening of the eustachian tube. And then if we added a little steroid, it was a little bit of a bonus on top of that. So then I was challenged at a conference, and I was challenged to treat acute otitis media. So that you cannot do in gerbils and mice. You just need a bigger animal, and you need a bulla that you can actually test. So we did this in conjunction with a laboratory in Boston, where they're quite famous for their studies on vaccination for otitis media in children. And it was the director of the lab really wanted me to fail. Like, he wanted to take the money and he wanted me to fail. So I showed him, right, Joe? And so I was like, well, I'm not going to fail. I don't know what to tell you. So we had chinchillas, and their coats are terrible at the end of experiments, which is unfortunate. But we had 39 chinchillas. We had 13 who only got the propellant. We had 13 who got the surfactant on the same day that we inoculated their bulla, which is their mastoid, with Haemophilus influenza, and 13 who got the surfactant starting on day 2 after inoculation. There was no difference between the surfactant 1 and 2 groups, so we can just look at them in combination. There was a significant difference between the non-treated group and the surfactant groups. And so we looked at tympanometry. We looked at quantitative culture, because the chinchillas are big enough that you can take off their bulla. You can look at their middle ear space. You can actually take a culture. You can put their bulla back on, glue them back together, and send them back to animal care. They do get labyrinthitis when they get bad otitis media. So unfortunately, that's a second factor we have to look at. And then, of course, the duration of fluid. So what we found was that tympanometry became normal or near normal much faster in the treated groups, and almost not at all in the not treated groups. So the first 2 bars are the surfactant groups, and the placebo bar you can see as the 3rd bar. And you can see that the blue and light blue were about 50% or more in the treated groups. We did not give antibiotics to these animals, and you can see that we had quantitative culture analysis, which was the same in all groups until day 12, which is about the time that the tympanometry normalized in a significant number of the treated groups. And you can see that the red line is that the bacteria were still highly culturable in the non-treated groups, and really fell off in the treated groups. And this is without antibiotics. So this is just with the normal clearance of opening the eustachian tube and allowing the infection to resolve itself. As I said, chinchillas spin around and they don't eat and they don't drink. And if they get labyrinthitis from their acute otitis media. So when that happens, the animal care stops the experiment and actually euthanizes the animal. We lost 9 animals in the placebo group. We lost 2 animals in each of the surfactant groups due to labyrinthitis. But one animal twirled for a day and then stopped twirling and started eating and did not have to be sacrificed. So this was a very, very significant difference between treatment and not treatment. And then if you look, the chinchilla has a single ossicle. You can see it over there on this side. You can see this nice, sort of, it looks like a giant malleus. They don't have 3 ossicles like we do. And so we wanted to see when they were dry, when they were moist, and when they had frank pus. And you can see that in the placebo group on the day 27, the majority were pus or moist, whereas in the surfactant group, the majority were dry with some moisture. So this was also very significant and related to the opening of the eustachian tube. So we showed that we could take care of this in a passive opening pressure in a normal animal, reduction of days of effusion in otitis media with effusion, and reduction of acute otitis media in a severe inflammatory condition. So we are looking to develop this for human application. So the human application would be the treatment of airplane ear or scuba ear, maybe avoid conductive hearing loss and ear tube surgery in serous otitis media, help us avoid antibiotics in acute otitis media, because right now the guidelines say watchful waiting for 72 hours and give antibiotics only as needed after that. And most people and most parents are not listening to that guidance still. And we're seeing a plethora of antibiotic overuse because of that. And then the other applications that I think are really very interesting as well is to prevent the complications of chronic eustachian tube dysfunction, such as tympanic membrane retraction, ossicular erosion, or maybe this makes us go from a 85–90% success rate surgeon to a 95 to 99% success rate surgeon in those chronic ear cases where we have to rebuild the ossicles and the tympanic membrane. And we keep worrying about when they're going to fall apart again. So I think there's a lot of interesting applications for this, and we've shown that we can get that agent there, and we can piggyback something else onto it if we need to, but as long as we even just get the surfactant there, we can get this to open up. So I think the future is the eustachian tube. It's the final frontier. We can boldly go where no otolaryngologist has gone before. And there's Dr. Crespi looking at the ear.
Thank you, Sujana. If you can stop sharing, this was terrific. I think if you stick around for the next group of speakers right after Dr. Hoffman, you will hear some of the pharma people showing variety of intranasal devices. The low-hanging fruit is airplane ear, period. Sometimes you see, your neighbor sitting next to you going like this as we're descending, right? It's an amazing opportunity, and hopefully this will clear the regulatory path soon, and we'll see that in the market.